Ladies and gentlemen, dear guests, esteemed colleagues, dear students, hello to everybody and welcome to Iasi, to our university, to the annual International Scientific Congress. Please, before all, to listen the Romanian anthem which is being sung. Thank you. Now, in the beginning, allow me to give the opening speech of the Congress to Mr. Rector, Professor Gerard Gitereanu. Thank you.
distinguished guests, dear colleagues, students, and members of the academic community. I would like to express my joy to welcoming you to the opening ceremony of the International Scientific Congress, Life Sciences Today for Tomorrow, organized by our university. There are almost 2 decades since our university organized this Congress together with the Ministry of Education and the Academy of Agricultural and Forestry Sciences, Bucharest. This year, our Congress will take place over 2 days into 14 thematic sections proposed by the 4 faculties within 380 participants from 12 countries. The program includes oral presentations, workshops, and poster sessions with the active participation of the professors, researchers, and PhD students from Bucharest, Cluj, Timișoara, Galați, and Iasi universities, as well as from the research stations and private companies. The challenge of this period is to use modern research methods to ensure the improvement of the level of agricultural productivity without any damages for the environment represented by water, air, soil quality, and biodiversity. In the current global context, agriculture must be adapted to climate changes by rehabilitating irrigation systems or applying modern technologies to the soil, such as no-tillage and conservative systems. Another challenge is food security, with organic farming on an upward trend, grown in the last decade from 2% of the total agricultural used area in 2015 till 42% at the end of this decade. Research also means increase of food quality, with new technologies leading to a circular economy, where nutrients and organic components of the soil have a central role. Last but not least, the livestock sector and animal pathology are the key to ensure the biosecurity of food in the farm-to-fork circuit. I invite you to discover during these days the news regarding the research in the field of life sciences and agronomy, together with specialists who agree to present their scientific papers and are ready to participate in discussions aimed to ensuring a healthy life in a healthy environment. Also, we are looking forward to the presentation of the main speakers from Justus Liebig University, Gießen, Germany, University of Córdoba, Spain, University of Naples, Federico II, Italy, and from Agri-Food and Biosciences Institute, Belfast, UK, and the University of Life Sciences, Timișoara, who have comprehensive and very interesting topics. And finally, I wish you all to have very fruitful meetings. Good luck, and I declare open the proceedings of the Congress. Thank you very much.
So going to plenary session, please we introduce senior scientist or akademischer Rat Christian Obermeier from Justus Liebig University, Gießen, Germany. I think he's present for 6-7 times to our congresses. The focus of his research is disease and pest resistance of crop plants, especially quantitatively inherited resistance against fungal pathogens and insect pests of oilseed rape. Research covers classical and genomic-based genetic approaches for dissection of complex threats. He is interested in understanding the molecular and metabolic networks and biological functions involved in qualitative and quantitative disease resistance expression in crop plants. Please, Professor.
Thank you very much. This is my 7th stay here. Thank you for inviting me over and over again. It's a pleasure to talk to you. I want to mention also that next year the University of Justus Liebig and University of Iași has a long-standing collaboration. Next year we will celebrate 50 years of collaboration. And I want to give you today an overview about the developing of insect-resistant oilseed rape. That's a very pressing issue in cultivation of oilseed rape, but it's similar in wheat and other crops. And I will give you an overview about the challenges we have, and then show you some of our results, how we're addressing these issues. So first of all, here's a survey of international experts all over the world about the top 10 most priority problems of pests and diseases. And in Europe, there are the cabbage stem flea beetle is position number 1. So in red, you can see the insect pests, and without color, the fungal diseases. So 5 of the most important ones are insect pests, and 5 in Europe are fungal diseases. And it's different in other continents like Australia, there's only 1 insect pest. But similar in China and Canada, some fungal diseases are a worldwide issue like Sclerotinia stem rot. But we have no cultivars with resistance against any of these insect pests in oilseed rape, and similarly in wheat and other crops. We have chemical control. So for fungal diseases, we have some resistance, monogenic partial resistance, some experimental resistances breeders are already using. For insect pests, we don't have that. We have chemical control, but chemical control is getting difficult because of the ban of neonicotinoids, for example, and more stronger regulations for using pesticides. So we have to do something about this. The reason that in the past no breeding efforts have been taken is that these insecticides were very effective and cheap. And on the other hand, that many of these insect resistances, or some we can find in experimental old varieties, are inherited very complex quantitatively. So they are different to tackle. In the past, it was difficult with the tools we had to use these. But, and in the last decade, it was even worse because the ban on neonicotinoids for seed dressings since more than a decade, and that many of these insect pests developed resistances against the chemicals which were still available to the farmers. So a breakdown of the efficiency of these insecticides. And also the political situation has changed, so we have sustainability goals now. And so that means that crop breeding, or breeding, becomes attractive the first time, actually, not again, but the first time. We are lacking 20 years compared to fungal diseases in oilseed rape, but we have all these new technologies nowadays, like high throughput, omics-based analysis, genomic sequencing, And so we're confident that we can now tackle this very efficiently. If we look at what kinds of strategies we could use to create insect-resistant oilseed rape or wheat or in other crops, here are some strategies or tools at the top. So we can use, on the right, genome engineering, for example. This is not licensed in Europe, but might be. We have genetic and genomic data integration like bioinformatics. We have on the left phenotyping by high throughput, camera-based drones, and so on. We have to develop indirect methods which are fast and can screen thousands of offspring in a short time, like spectroscopic methods, hyperspectral imaging, for example. And in the middle, we have interspecific hybridization. This is specific for oilseed rape. It means you cross two species, the progenitors of oilseed rape, turnip rape, and cabbage, to create a new Brassica napus again and introduce resistance this way. On the bottom, these are the breeding targets which we could tackle. Glucosinolates, for example, are a defense mechanism of oilseed crops against pests and insects. And I've shown on the left, this will be a short perspective because we know a lot about glucosinolates. They have their own problems. They are not a golden bullet, but because we know so much, it would be faster than some of the other things like tackling an improved microbiome, where a lot of research is still necessary. We have two bottlenecks in oilseed rape. One is that it was produced in the Middle Ages by crossing naturally crossing of two species, Brassica rapa and Oleracea, turnip rape and cabbage. And people just selected this because they could use it for lamp oil production at that time. So this was not very stable, but you could make this interspecific hybridization. So interspecific means between two species. And the other bottleneck is that since we used it as a lamp oil, we have bred to reduce anti-nutritive components of the oil, so we could use the oil as vegetable oil for human consumption. And these components are glucosinolate and russic acid. So we narrowed down the gene pool a lot and lost a lot of resistances on the way. Here this is shown. Double plus varieties have glucosinolates and russic acid in the seeds. Only one single cultivar is the ancestor of all our rapeseed today. So really genetic narrowing. And then Bronowski in the 1980s, another one which removed russic acid, and this also led to a further degradation of the genetic diversity, losing resistances. Now, interspecific hybridization is one idea to introduce again resistances. This you can do by crossing Brassica rapa and Brassica oleracea, diploid species. One has an AA genome, so diploid, and the other the CC genome, also diploid. And if you cross it, you have a tetraploid Brassica napus. But there are also other species in this triangle of view which can be easily crossed, like Indian mustard, Brassica nigra, black mustard, Brassica carinata. So this is one strategy to introduce resistances. On the left, you see all these species which are easily crossable because they're from the triangle of view. On the right, you have other Brassica species which we can also cross, but the offspring will be unstable and it takes years to introduce and stabilize them, to stabilize the chromosomes. So that is one strategy. Actually, this has been done, and here's a table showing the results for many different insect species on the left. In the middle, or in the first column, no Brassica cultivars existing have resistance to the pollen beetle, or to the cabbage seed pod weevil, or to the rape stem weevil. Sometimes you find reduced susceptibility, but if you look in the middle column, these are now the interspecific hybrids, you find no red signs, there are some more resistances. So we can try to exploit this. And on the right column, we have other species. In red and blue, the ones which are easily crossable. Sometimes for the pollen beetle at the top, we find something in very far away species like Synapsis alba, which are difficult to introduce the resistances against this insect into pollen beetle. The next bottleneck we have is the phenotyping bottleneck, that in the field it takes a lot of effort and work to look at the damages, to record them in a reproducible way. And people have been trying to solve this by using camera-based high-throughput phenotyping, for example. On the left, at the bottom, you can track insect activity on leaves of different genotypes, hundreds of them, with a camera, and by the activity, you can say that some of them don't feed, they don't like the genotype you are exposing them to or the leaf. And by this, people in Wageningen, for example, identified a gene for aphid resistance, which has a broad spectrum activity against phloem-feeding aphids. In the middle, you can use some proxies. You can use viruses, load insect with viruses, inoculate thousands of plants, and then do an immune test in yellow. This is a positive reaction for virus transmission, and this indirect could be used for resistance to the aphid. Of course, you have to figure it out in the next step. Or on the very right, you see a drone image from damage from the top. This is difficult because some insects are in the stem, some below the leaves, so it doesn't always work. But this is what we have to go for. If we talk about breeding targets, you could think about secondary metabolites which have a negative impact on the insects. For example, saponins. But saponins are not produced by oilseed rape. They are produced by wintercress, for example. This also has been tackled. It has been crossed with Brassicinapeus, but this is now going on for 10 years, and the offspring is not stable. This is very difficult. Or there are other components, but some of them need HPLC, gas chromatography, so very complicated, expensive technologies which breeders cannot provide in their environment. And then there's a knowledge gap because many of these components, like flavonoids, have also positive effects. If you remove them, that will have an effect on yield or other properties. So it's difficult to just go for secondary metabolites if you don't have identified one really good target metabolite. Glucosinolates, like I said, are long known. We could try this. This is known as the mustard oil bump. It's a system to defend oilseed rape, a natural system where the myrosinase is creating breakdown products if the leaves are chewed, and these breakdown products are active against insects. But in Brassicinapeus, we have specialized insects. They like this stuff. So they come and they metabolize it, they detoxify it. So if you change this, it will work against a few insects, non-specialized insects, but not against every insect. But what we urgently need is new genetic resources to search for resistances against pests. And we need these new high-throughput phenotyping resources to make progress, to test hundreds or thousands of offspring. And we need molecular markers and biomarkers like metabolites, glucosinolate profiles, for example. So we started a little experiment to create new novel genetic resources. These are 105 genotypes. On the right, you see some of the four. They look very different. Some are elite hybrids. It's like Arianos from KWS. And then we have resynthesized lines, so these interspecific crosses, R53, for example. We have exotics, and we have fodder rapeseed, so all kinds of diversity. And on the left, you see they are also deposited in a genetic tree at very different branches. So they are diverse, and these we tested. 38 old line varieties, 33 resynthesized lines, 11 winter fodder, 11 exotics, and 12 commercial hybrids as a control. Here you can also see that at a certain time point, 17th of April, not all of them are flowering, they're in a different developmental stage, which could also produce problems when looking at insect resistance. We planted them at 3 locations around, so you see Frankfurt here, and south of Frankfurt we have a field station, Groß-Gerau, and north we have Gießen, and we have 1 location close to Marburg. This is 135 kilometers apart, very different environments, 2 degrees warmer in Groß-Gerau, mild winters, and we put all these plants in replications in these 3 fields. And I will talk to you about resistances or different types of interactions against 3 of the insects which occur in Germany. The blue ones occur in winter or autumn. So 2 of them, the cabbage stem flea beetle and the rape stem weevil, will occur in winter. We can phenotype in about February. And then another one, which is a spring insect or summer insect, the rape stem weevil. We looked at more. To phenotype them, we sampled about 6,000 plants from the 3 locations, brought them home, 10 for each genotype from each plot, and then we looked at them. So the first thing is we dissected them. We cut off the stem to see if there are larvae inside and counted the larvae. This is number 1 here. We also measured the biomass. After we did this, we put the 10 plants after dissection into a box and kept it for 3 weeks in the greenhouse at elevated temperature, so over 18 degrees, to expulse the larvae which are still in there. This is called the Berlese method. From 1905, Berlese, an Italian, developed a method where he put soil into a box with a funnel, and from the soil he expulsed or drove out at high temperature the insects and counted them. We do this in these big boxes in the second step. But you can see here on the right the larvae of the cabbage stem flea beetle. It has 3 little legs and a black head. And the larvae from the winter rape weevil on the other picture on the right looks very different. It's bigger, it has a brown head. So you can distinguish them and count them. This is what we did for all the genotypes, 6,000 plants. And the results are shown here. So at the 3 different locations, we have a very different number of total larvae, so both larvae together. Weilburger Grenze, Rauschholzhausen, Groß-Gerau. The numbers here mean percent of plants with weevils. So 0.9 means 90% of all plants have larvae. So Groß-Gerau, the largest infection. If you look at the right and we distinguish them, we can see that most of the larvae are the rape winter stem weevil, which is shown here on the left, the adult and the larvae. So we are studying natural infection. It's always different, and this makes breeding, of course, also complicated. If I put this data together, so this is an average of the 3 locations, you can see in red here on the left some lines which always performed similar, always low percentage of infestation with larvae, total larvae. And on the right, you see somewhere you have a lot of standard deviation. So on every of the 3 locations, it was different. But it's reproducible for some of them, which is promising. And they come from all different types of brassica. So some are resynthesized lines, these new type brassicanapes. Some are exotics, some are line varieties, some are winter fodder, but none are commercial. So it looks like maybe we find something interesting. In the second step, when we put all these into boxes and expulse the larvae in a greenhouse, you can see here these were like 1,000 or more boxes for every plot, 1 box from 3 locations. And in the middle, you can, after 3 weeks, you can count the larvae per 10 plants as a bulk result. And if you plot this, it's similar. You have some varieties old line varieties, resynthesized and exotics, which show much less larvae per plant than the commercials. The lead cultivars are here shown in yellow. So there's one which is good, but all the other ones are pretty bad. We can do genetics with this kind of data, so we can do what we call a genome-wide association analysis to link the markers, 17,000 markers, with the phenotype and then find a chromosome which controls these differences in larvae numbers. And we found that this is reproducible across location, which is a very promising result. So maybe we can develop markers for that. But this is just 1 year experiment at 3 locations. So we have to repeat this and see if we can exploit this. Some loci are environment dependent. So if you have now these different interactions, it doesn't have to be resistance. Resistance would mean active stopping of the pest or the pathogen. So like in the picture below, you see that all the different insects will be attracted by different plants, big ones, small ones, and then within the tissue, there's an active mechanism stopping them. So, for example, chemical defense, glucosinolates, or cell wall. But it could also be avoidance, which would mean that the insects like the bigger plants and avoid the smaller ones. And because all our plants are very different in the field, in their development, maybe it's not really resistance, it might be avoidance. Avoidance is not so easy to use for breeding because we don't want to breed for small plants which don't produce seeds. We need something like resistance. There's also other types of avoidance, like the insect don't like the smell, the volatiles of the plants. For this, you could still breed. But we have to find out the mechanism to be able to do targeted breeding. And if I now correlate the biomass as a proxy for development with the resistance, or with the percent of infestation, this is what I find here. So I ordered the plants now from a low biomass, 4 grams on the left, to very high biomass, 65 grams per plant. And if there's a correlation, if the plants don't like small plants, then there should be something like this. You could also do the statistics. Here it doesn't look like there is a correlation between biomass as a proxy for development with the percent of infestation. And this also doesn't look like it's the case for different subgroups. So here, the resynthesized line, there's no correlation. Here, the hybrids, no correlation, but you can see it only starts in the middle because all hybrids have a high biomass. They start with 40 grams per plant. But the general biomass, small plants are still infected the same way as big plants. Also here, in these exotics. So correlation is non-significant at Rauschholzhausen. There's a low correlation of 5 or 6% at the other. But that might mean that maybe this avoidance shown in the picture, the escape because of the development, is not the reason for the differences we see. And so maybe we are lucky and we have some resistance mechanism. This is what we're investigating at the moment in more detail under controlled conditions with contrasting genotypes. And other people are doing this in Europe. This is a collection of new programs, or some programs which almost finished, on breeding for insect resistance, not on using CRISPR-Cas or other. There are many other projects. But as you can see on the left, in France and the UK and Germany, people focused mainly on two insects, the pollen beetle and the cabbage stem flea beetle, which are the biggest problems. And in Germany, Nadine Austel and colleagues working for 9 years on that, and they still haven't, they have found resistance in Eruca sativa, but the offspring, the crosses with rapeseed, are unstable. So it's a long, long story. We are now started a new project called Res Forst Res, or resources for stress resistance in Germany, focusing on 5 insects. So in this big project, we don't only deal with insects, but also with sulfur supply, because sulfur is a big part of glucosinolates and metabolites which are involved in stress resistance, like glutathione. Here, I'm involved with my colleague Benjamin Wittkopf from Justus-Liebig-Universität, Nadine Austel, Thorsten Meiners, Christoph Böttcher are plant ecologists, chemical ecology from Julius Kühn Institute in Berlin. Thorsten Will is a specialist on aphids in rapeseed. Michael Rosters, entomologist specialized in cabbage stem flea beetle. Björn Usadel is a bioinformatician. And Annelies Mason is an expert in stabilizing crosses with wild relatives of Brassicinapeus. And then there are 4 breeding companies involved: Limagrain, KWS, DSV, and NPZ from Germany. And as I said, we're also studying the interaction with sulfur because we want to create offspring which is resistant also under low sulfur supply and under heat stress. Not just insect resistance, but stable in a different low cropping intensity environment. In this, we tackle these 5 different insects: the cabbage stem flea beetle, the cabbage root fly, the green peach aphid, the rape winter stem weevil I talked about, and the pollen beetle. And we use new phenotyping technologies like drone-based hyperspectral imaging, and we do this in the greenhouse and in the field, and use all these different types of genetic resources I showed you. Of course, there are also a lot of metabolomics involved to find new biomarkers for that. In summary, what we found until now is that the germplasm set we tested is suitable to identify interactions, different types of interactions, and we just started to investigate this in more detail. And some of these older exotics and recent designs show promising interaction, maybe even resistance, which can be putatively used for breeding. On the bottom, I show you another insect which looks similar, a weevil, but a summer-type weevil, and we see very different percents of it creates this S shape of the stems you can see in the field. And this is 2 to 52%, so very big differences which can be used for genetic analysis. So in summary, I think that in the next 10 years we can deliver some material to breeders which they can use for introducing it into their material. And the key is to combine old and new innovative high throughput methods. You can see on the left, genomic resources are very important to find new resistances. You should use new sequencing technologies, create pan-genomes of all these, so sequence all your material. You can use this bioinformatics data to map resistance gene and characterize them. You can use CRISPR-Cas to do functional analysis, to shut them down and see what happens. In resistance breeding, we can create these interspecific hybrids because they seem to be useful, and we have to develop high-throughput technologies, non-invasive technologies to do this. With this, I come to the end. I want to thank people involved in Gießen. Tobias Esch is a master student who was counting many of these insects, larvae, did a lot of work. Benjamin Wittkopf, my colleague, Lennart Roscher-Erich is a new postdoc in this new Race for Stress consortium. And then the other people from this consortium, Michael Rosters from Göttingen, Nadine Austel from Berlin, then Thorsten Will from Quedlinburg, Björn Usadel from bioinformatician from Heinrich-Heine-Universität, Annelies Mason from Bonn, and the 4 breeding companies involved. And I also thank the funding agency, the Ministry of Research, Technology and Space in Germany. And with this, I would like to thank you for your attention, and I'm happy to answer your questions.
Thank you. Thank you, Professor, for your presentation and also for your colleagues. And now, if anyone wants to put a question, yes, please.
Thank you very much. So my name is Julian Gabor from Iasi now, former student of Gießen. Christian, I have two questions, and it's related to the glucosinolates pathway or the glucosinolates content. We know that it's a natural defense mechanism in plants against insects. But as you said at the beginning, you don't want it in seeds. Do you think it's feasible to increase the glucosinolates content in leaves and stems for different insects, but also keep it low in seeds so you don't have the problem of selling the seeds at the end? That would be one of probably the challenges of this breeding program. And another thing you also mentioned at the end, that it takes up to 10 years to develop a new resistant variety or tolerant variety, let's call it. Would you think that maybe CRISPR or new genome technologies will help in this regard to pinpoint and target specific genes that would give resistance or tolerance to different insects, not especially for flea beetle, so in general for insect resistance? Thank you.
Okay, so the first question on seed glucosinolates and glucosinolates in stems and leaves. In the past, breeders were strongly selecting for low seed glucosinolate, which also led to a decrease of glucosinolates in leaves and stems. And this is one reason why insects now can like to eat the leaves. You can decouple that. There are transporter molecules, this has been also shown by transgenic approaches, which have been knocked down, and then you only have high seed glucosinolate and low leaf glucosinolate. You can also tackle this by breeding. You have to know Brassicanapeus has many different copies. It's a polyploid or tetraploid. So for any gene involved in the glucosinolate pathway, there are 8 or more genes, and you have to characterize them. Some are tissue-specific to know which ones to knock out or target by breeding. But in principle, this would be possible to decouple that and create rapeseed with high glucosinolate in the leaves and stem and low in the seeds. But the trick is not what the total glucosinolates have to be changed, but the profile. So specific combinations of glucosinolates, because as I said, there are specialist insects which like high total glucosinolates and certain glucosinolates. And you have to find a combination which works against many insects. And that is why it's not so trivial to do this. Even we know it's a natural defense system, glucosinolates. And the other question was on CRISPR. Yeah, we can use CRISPR. The problem with CRISPR is we need to know the targets. If we don't know the genes we want to knock out, then CRISPR doesn't help. So first of all, we have to do all this genetic stuff, find the genes which are really involved, which has a big effect, to design CRISPR RNAs to knock it down. And the other thing is that it's not licensed now in Europe. You can do it in the US or somewhere else. Maybe this changes in the future, but in the meantime, we have to try to manipulate these pathways by traditional breeding, by Mendel.
Congratulations, very interesting research. My name is Marga Grădilă. I work of Institute for Plant Protection Bucharest. I am responsible for a laboratory of testing pesticide, and you see in your slide aspects regarding the neonicotinoid, and your research respond of these aspects. Dear Christians, in Romania, it's a big problem. It's a major contradiction regarding the neonicotinoid. The farmers say this product is very necessary for higher production. The scientific research say this product is not toxic for bee because it's applied for seed treatment. Yeah. And in this context, my question is, which is your opinion regarding the subject?
Okay, so I'm not a specialist for chemical protection or reduction of the efficiency by evolution of the insects against these pesticides. But in my opinion, I'm a breeder, we just have to react to the political facts. And we have to just try to find alternatives, because it's not likely that the situation in regulating pesticides, and of course there's discussion if they're bee-friendly or not, will change. This will continue, and we will have less chemicals with a lower mode of action, and resistances will develop against these insecticides based on how this is practiced nowadays. So we have to find other ways, and one way is breeding. Of course, both should be done in parallel. We should find out which pesticides are safe for bees and other insects, and we have to have a management of insecticides. So we should educate the farmers not to use always the same insecticides over and over again, because resistances are developing fast. There's no way of stopping them. And in the pollen beetle, some of them don't work anymore. From 2007, pyrethroids, 2015, they found 7% of beetles where it didn't work, and in 2017, 94%. So this is just what is happening. Sorry, I cannot answer specific for this product, but I think breeding there's no alternative to breeding.
Any question, please? If not, we want to thank Professor Christian Obermeier for his presentation. Thank you, Christian. And now we follow with the second presentation from Associate Professor Dr. Jaime Moreno Garcia, an Associate Professor at the University of Cordoba, specializing in industrial fermentation and biotechnology. He is the head of the Vitanol Research Group, an active member of the Fulbright Commission in Spain, and an associate editor for the Q1 journal, Microbial Cell Factories. Dr. Moreno Garcia has earned 7 national and international awards for his teaching, research, and entrepreneurship. He is currently co-principal investigator on a 800,000 scientific project funded by AFRI, so Agriculture and Food Research Initiative, from USDA. The title of your work is Fungal Pellet-Based Cell Immobilization for Viniculture and Bioethanol Production. Please.
Okay. Good morning, everyone. Thank you very much, all the organization committee, for this invitation, and especially Julian and Jana Abur. So my presentation today is titled, as it was said, Fungal Pellets-Based Cell Immobilization for Viniculture and Bioethanol Production. Okay, in here this is outline of what I'm going to present. First, um, the pointer. Is the pointer? Ah, okay, okay. So first, I'm gonna briefly relate horticulture with enology and bioethanol production. Secondly, I'm going to talk about the fermentation optimization, how we can improve this bioprocess. I'm going to explain one way of improving it, like through cell immobilization system. I'm going to define what is it. Also, I'm going to focus on filamentous fungus pellets as a cell immobilizer, what have been done so far, and what are our current projects. So, as we all know, from the grapevines, we can obtain mainly four different products, which are the table grapes, the wine grapes, the vine leaves, and the grafted vine plants rootstocks. If we process the grapevines, we can obtain wine, as we all know. And this is a product with a much higher value. Also, from the sugarcane plant, we can obtain sugarcane stalks, cane juice, bagasse, and bagasse. From the cane juice, we can obtain refined sugar and molasses. And if we process molasses, we can obtain bioethanol. And in here, there is also a increase in the value from this product. What these conversion processes has in common? Well, there is one step in common of these processes, which is alcoholic fermentation. Alcoholic fermentation, as you all know, is the conversion mainly from sugars to alcohol and carbon dioxide. Also, it is this carbon from the sugar is transformed in biomass and other metabolites. And we, in order to improve the quality and the productivity, or the productivity of the fermentation process, and the quality of the final product, there are many different techniques. Among them, there is one that is called cell immobilization systems, which I don't know if you heard about it before. Well, cell immobilization system is a technology which aims to confine cells which are naturally in a suspension in a single region, keeping their metabolic activity. So why do we need that? Why do we do this? It increases the cell densities in a fermenter. It also increases the process control, the cell protection, and it's, the same cells are easily reutilized. So with these features, we can improve or we can increase the productivity of fermentation, and also we can, with the control, we can also improve the quality of the final fermentation product. Of course, there is also some cons, some disadvantages of this system as any other technology, which is costly, and there is a limitation in the mass transfer. So I mean like the rate of the substrate coming into the cells and the rate of the product released to the external medium. In winemaking, these systems are not new. Okay, they have been tested and they are already implemented in industry. I know that we don't know that much about these systems, but they are already used in the industry. For instance, in winemaking, alginate pits are used for sparkling wine production. And also there are some ceramics that are used also to produce wines. As I show in here, there are different types of material that can be used for cell immobilization, natural, organic, and inorganic. and they all have their pros and cons. For the bioethanol production, there are also like many different systems used for mainly to perform continuous fermentations, and again, the most conventional ones are alginate and the ceramics. Okay, this is a little weird. Okay, so my research group is focused on one of these immobilization systems that use fungal pellets as the carrier. The fungal pellets are little spheres that are produced when you grow certain filamentous fungi under agitation. They form these spherical shapes. And depending on the conditions, you can make them hollow. So we have a technology that we immobilize the cells inside these pellets. So they need to be a pelletizable filamentous fungus, and you can immobilize any type of microbial cell. For instance, bacteria, archaea, yeast, microalgae, or a consortium of several of these. This is how they look like. I don't know if you got this, if you work in microbiology, if you got in your flask this kind of shapes? No? Maybe from contaminations, but okay. So we started, yes, we discovered this as a contamination in one of our flasks, but instead of getting rid of them, we studied them. And since 2004 until now. So, so this is how they look when you look at them under the SEM, scanning electron microscopy. You can see the yeast cells that are attached to the fungal hypha. They are actually quite bonded to the hypha, as we can see with these arrows. And when they release from the hypha, they leave like a scar in the hypha. So you can see in these black arrows. Under the TEM, transition electron microscopy, you can see like the differences between the two types of cells. In the bottom, it's the hypha, and the above one is the yeast. And there, it seems that there is a polarization in the structure of the organism, and we can see like vesicles towards the hypha. So there is a metabolic interaction between these two organisms. So why do we use this and not other microbial cell immobilizers? It's a food grade, okay? So that opens a door for food purposes, like winemaking. It's biodegradable, so it aligns with the sustainability processes and products. It increases the mass transfer because this is a matrix of hypha, so it's a porous material, and that allows like the easy like substrate and product release and income. And if you have the possibility, and this is so important, to keep both microorganisms alive, so that there could be syntrophic metabolism. That means that when they are together, they can produce a metabolism that when they are by themselves cannot. So this could be so important for secondary metabolite production, like as we all know. But we have a problem, that the cell retention, okay? What did we do so far? We tested this system in winemaking, for winemaking application. And as we could see in here, my colleagues published a paper that was, the main aim was to accelerate and control the grape mass fermentation process. And this is a this is a fermentation monitoring that, that shows the CO2 release. And as we can see in here with this arrow, the fermentation performed with this system was shorter than the one with the conventional method using free yeast. Okay, so this is an advantage. Also, we observe higher amounts of acetaldehyde and isobutanol when using the pellets. In another work, we studied the interaction between the yeast and the fungus, and we observed that during a fermentation, the fungus died. And this is the image is a fresh system with the yeast and the fungus without before fermentation, and the C1 is after fermentation. And in the C1, we can just see that the yeast grow on a YPD Petri dish. Then we investigated the immobilization of osmotolerant yeast for the production of sweet wines, because as I said before, the fungal pellets provide a protection against different stress, as osmotic stress. And we could see here that in this paper, there were higher scores by blind tasting of the wines produced by using the pellets system versus the free yeast. Also, we assessed the system producing sparkling wine and we compared with other formats with free yeast. And we could see that the reading time was significantly reduced to less than 2 minutes compared to several days for free cells with bentonite. And also in here we can see the foaming properties, like the foamability and persistence was similar or even better than when using free yeast. Also, the purpose in this paper, which proposed was to improve the sweet wine production, the system was show a satisfactory operational stability during 5 repeated fermentation. So you can reutilize. We tested the reutilability of the system, and we could see that it was like functional for 5 times. Also, we studied the effect of 2 strains on the aroma composition and the sensory quality of sparkling wines, and the yeast immobilized in the fungal pellets generally showed the highest values for most of the aromatic series and best scores for aroma quality in sensory analysis. In here, well, this was my first paper related to this system. The ones before were like my colleagues. And in here, we wanted to go further to try to see what works better, what yeast work better in this immobilization system. We had the initial starting hypothesis that flocculant yeast and yeast with biofilm formation capability will be like good candidates. And we could see that biofilm forming yeast, like those that are used for maybe sherry wine production, perform so much better than those that don't have this capability and the flocculant yeast. Also, another test on sparkling wine observed that there were specific aromas that were improved or increased, like the balsamic one, as we see in here, in comparison with the free yeast. In here, I was also part of this paper. We wanted to avoid or to prevent the problem of the cell retention. So what we did is to add a layer of alginate to the system, like trying to avoid the cell release from the system. What we could see, so this is what we did, like the alginate coated biocapsules. What we observed is that we were right. We could lower the cell retention, but at the same time, we lowered the mass transfer. So you have to choose one or the other. Yes, the ethanol content was a little lower when you compare with the uncoated system, as you can see in the orange bar. And this paper purpose was to optimize the red raspberry wine brewing. The immobilized yeast, they showed that the immobilized yeast had a higher fermentation rate, as we can see here. They obtained up to a 15 degrees volume ethanol content, and they could, using this methodology, significantly preserve active components like anthocyanins and polyphenols. Also here, this was a work in collaboration with UC Davis in United States, also with the objective to prevent cell leakage. Instead, we changed completely the methodology. So we, first, we obtained the pellets, we inactivated through autoclave, and then through vacuum, we encapsulated the cells inside. What we could see in this new methodology is that we increased the productivity of the capsules a lot. Also, we improved we lowered the cell leakage and improved the cell retention. And also we tested the winemaking application with this new methodology, and we observed that wines were positively evaluated. Also, we produce sherry wine that are very famous in south of Spain, and we could keep the ethanol content during the biological aging for the time tested, that it was 3 months. So this is interesting for the winemakers because they might not need to fortify the wine that will be costly. Also, we got the highest score using this methodology versus the conventional one. Also, with the purpose to improve the aroma quality, polyphenol profiles, antioxidant activity, and color stability of Merlot red wines, these authors observed that the biocapsules, the system with the fungal pellets, increase desirable esters like ethyl 9-decanoate and ethyl 1,3-methylbutanoate. They also use for the first time a fungus, which is Alternaria alternata, and they could observe that they work well with this system, this fungus. Then we assess the impact of the immobilization with immobilizing non-Saccharomyces yeast, and we also obtain higher score than the conventional fermentation, and increase the total acidity and lower the ethanol content versus the free yeast systems. Also, we try to customize different wines with adding through sequential fermentation, using different pellets with immobilizing different yeast, and like trying to target a specific metabolite, and we could obtain it. We target 1,1-deoxy ethane, and we could increase with this methodology. So that means that you can use this technology to customize your wines and improve the quality. And also, these researchers tested and confirmed that the yeast cells are more protected towards stresses by sugar content, ethanol, pH, and temperature in this article. And in terms for bioethanol, we also tested, and there could be we tested the reusability, and we could observe that the yeast immobilized were functional for 7 times. And yes, and also the highest productivity was obtained in the second batch. Also, we tested the alginate coating in here, and as we observed for winemaking, we also observed the same thing. The cell retention was improved, was reduced, was increased, but the mass transfer was compromised, as we can see in the total ethanol content. And finally, a very recent paper obtained with this system very high ethanol content and very high yields with using this system. Okay, so what we can say here is that this system is good for these 2 applications, even the science supported, as we could see in here with these scientific papers published in these known journals. Also, it improves over the conventional suspended systems and also alginate for both applications. So I'm going to tell you what we are doing right now and what I think that it should be done in the future. What we are doing right now is trying to produce wines with reduced alcohol content, keeping the quality or even improving it. And we are doing this through sequential fermentation, first non-Saccharomyces yeast, immobilized, and then Saccharomyces yeast. And with bioethanol, we are trying to produce second-generation bioethanol, which is coming from the agricultural residues, trying to lower the price because currently is high in comparison with the first-generation bioethanol. And I think as we could see the potential of this technology in a lab scale, next step would be a techno-economic analysis, also LCA, which is life cycle assessment to see if it's sustainable or not. And if we have a check for these 2 analyses, go for scaling up. So with all of this, I want to thank you for your attention, and I am open to answer all the questions that you might have. Thank you.
Thank you, Jaime, for your presentation. And now, if you have questions, please, immediately.
Thank you very much for your presentation. It's not my topic for the wines. I'm a consumer. I mean, with moderation.
Okay.
This is very good. My question is, for the future project bioethanol, what would be the impact for the environment with your technology? I mean, it's a good one, it's a bad one, because we know that bioethanol, for example, some cultures in some cases has had a negative impact on the environment.
But talking about bioethanol in general, or using this technology for...
Because I don't know
Okay.
about this technology.
In a sustainable context, I think that it will improve the sustainability because this, as other immobilization system, you can reutilize the same yeast, so you will So the same material you will be reutilized. So, and also it's biodegradable, so it's not like other inorganic material that you use it and you release it to the nature and they are so difficult to degrade. Also, we believe that it's more efficient or with the use, the productivity will be higher than other conventional systems because, as I said, it's a matrix, it's a porous matrix, and when you perform alcoholic fermentation that the gases, CO2 gases are evolved, the CO2 can be easily released from the system because it's porous. But alginate is not porous, it's a gel, and it can get like even broken as and released to the external medium. So yes, reusability, more biodegradability, and more efficiency for.
Okay, I have a question. I'm Valeriu Cota from this university. You know. Okay, it's a very interesting presentation and tools which you can play with the yeast. And I saw you can use different kind of yeast, Saccharomyces or non-Saccharomyces, so the purpose are different. The question is, how much yeast do you use for 1 hectare or whatever? Because you have the natural yeasts in the liquid, must or whatever. Mm. And you have to fight with the natural yeast.
Yes.
If there is a long starting of the fermentation, you can lose the battle. Thank you.
Okay, yeah, thank you. Thank you, Valerio. The amount of G cells is, we use the conventional amount for industry that is 5 million UFCs per milliliter. With that very high density of cells, when you inoculate your grape must, which has been already treated with sulfur dioxide or tartaric acid, it usually works. I think that almost if you have to make sure that your yeast are active in the middle of the exponential phase. Once you have that knowledge that the yeast are active and you inoculate a very high amount, I think that you have a very high chance to conduct your own fermentation and not the spontaneous fermentation that comes from the autochthonous microbiota. Thank you.
Yes, other questions? Please.
Thank you very much. A very, very interesting presentation and very complex from a lot of points of view. I actually have a question about, you mentioned there the wine quality, and it had an asterisk. What exactly is it, the wine quality, the correctness of the wine? Is it the wine quality as measured by the OIV in wine contests? What exactly do you mean by wine quality?
Wine quality? Well, as I previously said when I introduced the emulsion system, you can, like this system, this technology, helps you customize your fermentation. So wine quality, I mean, if you want a certain metabolite that could be antioxidant or provide organoleptic property that you want for your market, for like glycerol, acetaldehyde, you can select a yeast that specifically produce that compound under certain conditions, you inoculate, and then you remove whenever you want. So this is what I mean with quality. Yeah.
Thank you. Please. Dear Jaime, I have a question about the carrier. You mentioned Alternaria alternata as the first carrier. After you acknowledge, do you know if exist other filamentous fungi which can be used as a carrier
Well, you mentioned alternator, right? Yeah, that's, we didn't publish that paper, okay. But I think that is a pathogen, of a plant pathogen, right? It's a tomato or something. I don't know why they choose that one, but yes, there are. I have to say that not all filamentous fungus can be used to produce these pellets, okay? But yes, Penicillium chrysogenum, Aspergillus oryzae, that is the one that we use because this is used for sake production and it's a food grade. And also Aspergillus niger is also pelletizable. This is not. Has mycotoxins, so I would never use it for food purposes. I would use it maybe for bioethanol or something like that. But yes, there are some, but not all of them are pelletizable. Yeah.
Thank you. Is there anyone who want to ask? If not, thank you again, Professor Jaime Moreno Garcia, for your presentation. And now the third speaker. I invite Professor Gaetano Oliva. Hello, Professor. It's Professor of Veterinary Internal Medicine, Department of Veterinary Medicine and Animal Production, President of the School of Agriculture and Veterinary Medicine, University of Naples, Federico II, Italy. Professor Gaetano Oliva is a full professor at the Department of Veterinary Medicine and Animal Production, where he has been active since 1987. His expertise lies in veterinary internal medicine, with a special focus on zoonosis and vector-borne diseases, particularly leishmaniasis. He has served as Dean of this Department of Veterinary Medicine, Director of the University, Veterinary Teaching Hospital, and since 2023, President of the School of Agrarian and Veterinary Medicine at the same university. He also leads the Regional Pharmacovigilance Center for the Campania region and serves as President of the International Scientific Group LeishVet, contributing to global guideline for the diagnosis and treatment of leishmaniasis. Professor Oliva is member of the World Small Animal Veterinary Association, One Health Committee, and collaborates with numerous international institutions. He has been invited speaker at major international conferences like World Association for Advanced Vet Parasitology and ICOPA, its International Congress of Parasitology. Next year will be the 16th Congress in Montreal, Parasites in a Changing World. It will be the theme of this Congress. So the paper is Animal Leishmaniasis. It is time for One Health approach. Please, Professor. Thank you.
Thank you so much. Thank you for the invitation. Thanks to the rector, the dean of the Faculty of Veterinary Medicine, and Professor Basile Vulpe for the warm welcome. It is an honor for me to be here, and thanks to all of you. My topic is about leishmaniasis, but just I would like to give you a very short presentation of my school, because my school has two departments, one of the agricultural sciences and the second veterinary medicine. We belong to the University of Naples, Federico II, that is the oldest public university in the world. Was founded in 1224. So the last year we celebrated 800 years of history. And the school was founded in 1846. You can see that Department of Agricultural Science since 1873. And as a veterinary medicine faculty, we were established in 1798. And actually we are in two historical buildings, one the Palace of the Kingdom, Charles III in Agrarian School, and the Monastery of Sant'Angelo alle Croci, the School of Veterinary Medicine. So please come to Naples to visit our school, because it is not only a scientific collaboration, but also a very nice touristic visit. So go to leishmaniasis, that are a group of diseases, you know, that are causing bad protozoa belonging to the genus Leishmania, that affected humans and animals, and are conditioned by change of the environment. So we are in a context of One Health approach. And here you can see the distribution of the 20 species that affected the humans all around the world. Leishmaniasis is the second important protozoal disease after malaria that affect the humans. So it's very, very important, and you can see also that it is mainly distributed in humans in poor regions, or it is defined the disease of the poorest of the poor. So it's time to fight against this important disease. In red, you can see the species, Leishmanium infantum, that is the dominant in Europe, and we have this species both in humans and in animals. My topic will focus on the need of change of paradigm, because sometimes leishmaniasis is approached as a human disease for which animals are just reservoirs. But now it's time to change paradigm, because you can see here that the animals and humans may act as a reservoir both for the spreading of the parasite all around. So we need to avoid this kind of classification. This is a paper that appeared 10 years ago, not 1 century ago, 10 years ago, and again, you can find in literature 2 kinds of cycle. One is defined anthroponautic, where the man is the responsible for the cycle, and the zoonotic cycle, where there is an animal reservoir that is a passive reservoir for humans. So we have to change this paradigm, because the only actor, real actor, is the insect. That is sandfly, is like a mosquito, that may transmit parasite to humans and animals. And both human and animals may act as reservoir. In humans, we have very good classification, clinical classification of the disease. You can see the visceral leishmaniasis, the cutaneous leishmaniasis, that is the most important all around the world because of its diffusion, and the mucocutaneous leishmaniasis, that is a disfiguring disease that is caused by many species, and the Leishmanium infantum also may cause all the 3 clinical expressions: visceral, cutaneous, and mucocutaneous. And in domestic animals, we have a clear definition of the disease in dog and in cat, because during the last 10 years, we know that also cat may be affected, both not only could be infected, but also could have clinical expression. And in these animal species, we have usually the generalized leishmaniasis that we call zoonotic visceral leishmaniasis, and some cutaneous or localized mucocutaneous form. So you can see in many dogs and cats just cutaneous or mucocutaneous localization. Here some picture from literature of the three forms in humans. The visceral leishmaniasis that most affected children in Africa, actually, in sub-Saharan regions of Africa, and it is fatal in more than 95% if not treated. The cutaneous leishmaniasis and mucocutaneous leishmaniasis that are severely disfiguring for people and mainly for children, because cutaneous leishmaniasis is not fatal, but it can cause scar in children that are persisting for the whole life of these children. Here, there are my picture. You can see Leishmania infantum may cause just cutaneous lesions that are sometimes also spontaneously resolving. And Leishmania major, here a picture that I did in Tunisia in children. And you can imagine how disfiguring could be during the life of these children. Some picture for dogs. You can see in the middle that we can have only cutaneous lesion. Here we call papular dermatitis in the ear of this dog, till severe expression that are also in dog that are fatal if not treated, and many times could be fatal also if treated. Why this picture? Because there is a clear relationship between the clinical expression and the severity of dog as a reservoir. So if the dog is sick, it is more dangerous as a reservoir because it's full of a parasite. So one of the messages is that we have to protect dogs with the repellents, also if yet sick, because many owners think, okay, my dog is sick, or I don't need to put repellents to avoid the bite of insects, and we must protect sick dogs and infected dogs because they act as a reservoir in urban and peri-urban context. Here the distribution of Leishmania infantum all around Europe. You can see that also in Romania we have clinical cases of human and also canine leishmaniosis. Not all are autochthonous, but there are many autochthonous cases, mainly in the border with Bulgaria. So it is clear that there is a continuous spreading of leishmaniasis from South Europe to the North Europe. And then I will show you some picture of what happened in Italy and where we were till 30 years ago and where we are now. So it is clear that my message is that also in Romania you have to be prepared, because with climatic change it will be for sure the localization of sandfly insect also here in Romania and the expression of a clinical form in here. You can see also other species that are surrounding Europe. Here, Leishmania tropica, Leishmania major, and interestingly, in the low part of the screen, Leishmania donovani. Leishmania donovani is the most important visceral species out of the Europe, is mainly in India and in Africa, and it appeared in Turkey in many cases, and we have also in Italy and in other parts of Europe the isolation of hybrid. One of the new knowledge that we have on leishmaniosis is the possibility of a creation of hybrid species. So the interaction between two or more species of leishmania that may exchange genetic material, and we don't know what is the pathogenetic significance of the hybrid in human and in animals. We are conducting many studies about that. So the appearance of new species all around Europe, for sure, these species will be introduced in the near future years. And why one not approach? Because we have many socio-economical factors, political and migratory influences, and environmental risk factors. Actually, we are studying all these aspects because they are, for sure, they conditionate the appearance. We know for sure that after war, there is an dramatic increasing of human cases of leishmaniasis all around the world because of the people are poor, in poor hygiene state after the war, so there is an increasing of immune imbalance, there is an increase of appearance of insect, and this is an increase of animal reservoir around this situation. So we have the spread of leishmaniasis. And who is the main actor? The phlebotomy sandflies is an insect that is the only insect that is able to transmit leishmaniosis. And this, we have many demonstrations of, we can find the DNA of leishmania also in ticks or in fleas, for example, but these insects are not able to maintain the complex cycle of the parasite till the new form, the new infective form. Because you know that in vertebrate, we have the amastigote form that is not flagellated form of the parasite that must develop in the sandflies for 8 several stages till the metacyclic stage of the parasite that is flagellate, and it is the infective form. And in the female of sandflies, the female sandflies is the only insect able to transmit this parasite. We know also that during, for example, at the beginning of 2000, during the appearance of the HIV era, we had all around Europe an explosion of human cases of visceral leishmaniosis because of the immune compromised patients. But actually, we have also many cases in long-term immune suppressive treatment in people treated by long-term steroids or other immune suppressive treatment, or in diabetes, or in other chronic diseases. You can have the appearance of this parasite. It is important to recognize that in the region of Europe where leishmaniasis is present, we have many people that are infected but asymptomatic. And you can have, after 10 years of asymptomatic infection, you can have the appearance of the disease because of, for example, an immune suppressive treatment. So it is an opportunistic parasite that could have, but it is important also to recognize that this parasite is able to do clinical expression also in immune-competent patients. We studios, the authors of the beginning of 9th century called Leishmania infantum, because at the beginning they believed that it was only a disease of baby. But we know that it can be in all patients on all age. Sorry. And very interesting, the new knowledge about this disease. The example of Fuenlabrada, we are in Spain, near Madrid, 25 kilometers from Madrid. You can read that in Spain, from 1997 till 2011, we had 8,000 hospitalization from Leishmania, but just in 2009, there was a significant increase of hospitalization just in one hospital or two hospitals of Madrid. There was a lot of visceral and cutaneous cases by Leishmania infantum. And the physician had not explanation for that. And there was a study all around this hospital because many people came from Fuenlabrada, were living in Fuenlabrada. So they checked for dogs, they checked for cats, but the seroprevalence in these two animal species was the same over the previous years. So they also identified that many people that were in the hospital had the habitude to go in these two parks near the city of Fuenlabrada that were full of hares that were there since 20 years, and they checked in the hares if they host Leishmania, and they found that hares had a lot of Leishmania parasites. So they were the reservoir, and now we know that also hare, but also rat, also foxes may act as a reservoir. So we have many, many animal reservoirs. Dog is a good reservoir because there is the disease and because we have dog in our houses. So it is dangerous as a reservoir because there are a lot of dogs together with us, but we have many, many animal species that may act as a reservoir. And new studies, we are doing new studies on the possibility that the dog is a reservoir not only for Leishmania infantum, but also for other Leishmania species. You can see here some picture from my colleague and friend Gad Banet for Leishmania major, for Leishmania tropica, or other in Brazil for Leishmania brasiliensis. And I had the possibility to publish two different papers, one on Leishmania donovani. I participated to a study in India to check for the dog as a reservoir of Leishmania donovani. And we also found for the first time in Tunisia that dog may also host Leishmania major in this paper, but we submitted a new paper in which we confirmed the presence of Leishmania major in dog in Tunisia and also Leishmania tropica. So it's clear that the dog may host also other Leishmania species, but the clear role as a reservoir of these species should be better investigated. Here some picture from Tunisia and for India is my topic of interest in research, leishmaniasis is a very nice topic because you can visit many countries that are very fashionable from this point of view. But I like to do field study, to stay together with the people and colleagues. And what we need to say that the dog is a reservoir, the only examination we can do is the xenodiagnosis, for which you put the dog in a cage, an infected or sick dog, and you release female of sandflies for 1 hour. Then you have to collect again sandflies to check if they were able to take parasite from, because the only demonstration of the PCR positive is not enough to demonstrate that there is the development of the infective form of the parasite into the insect. Why we have to be ready to the spread of leishmaniasis also where insects are not present? Because we know for sure that there is a possibility of vertical transmission, so from the mother to the puppies, venereal transmission, but also for humans, organs transplant transmission, and also blood derivatives transmission. So the vertical transmission is very important in not endemic country, because you here in Romania could import infected dogs that could give newborn with the infection. So it is one of the situations that we are assisting in north of France, in Germany, and in other European countries where sandflies are not always recorded. What happened in Italy? We were here 40 years ago, and you can see the spread of leishmaniasis all around Italy during the last 40 years. North Italy was considered not endemic when I was student. When I was student, leishmaniasis in my book of infectious diseases was described as a tropical disease, And now the North Italy is considered endemic. We did a study during the 2021 in which we recorded 57 new autochthonous cases in municipalities of North Italy, and you can see that where the blue triangle, there is the new location of autochthonous cases of canine leishmaniasis. So dogs that were living there since they were born there, and we recorded also the presence of the insect here in the right border is Trieste, so is a continental climate. In Valle d'Aosta, we have near the France, we have autochthonous cases with the record of sandflies up to 1,000 meters in Alps. So it is important to recognize that something is happening. And in Tuscany, actually, we have an emergence of pediatric cases for human leishmaniasis. So there is a continuous spread of leishmaniosis. This is a paper we submitted 15 days ago with the new estimated canine leishmaniosis incidence in Italy. You can see that we have no provinces out or with no leishmaniasis. So not all are autochthonous, but all around Italy we have canine leishmaniasis. And we were here 40 years ago. I am going to conclusion. What about prevention treatment? We have to prevent the animal reservoir. We have to apply repellents. We have vaccine in Europe for canine leishmaniasis, but the vaccine is not able to prevent the infection, is only able to prevent the clinical severity of the disease. And we have also to protect and treat infected and sick animals. In India and in Bangladesh, there was a very strong effort against human leishmaniasis by using repellents in the habitation of people. The vaccine is not available. Actually, Bangladesh is considered free or at the limit of free of human leishmaniasis, and also India is improving in this situation, but Africa actually is an emergence. And also Brazil, in the poor part of Brazil, is a very important site for human leishmaniasis. And as the Naples University and Istituto Superiore di Sanità of Rome, we are proud we are participating to one Horizon project, the CLIMOS project, that is studying the sandflies population and the climatic change, because sandflies and sandflies-borne diseases may act as a sentinel for the climatic change. So, what is my last message for today? That we have to control leishmaniasis as an integrated approach by acting mainly against the vectors, but fight the poverty, fight against global warming, fight against deforestation, and do everything to save the environment. That are the best strategy to fight against leishmaniasis, and consider companion animals just in the loop as innocent bystanders. And save the date, because as a LeishVet group, we will celebrate next Congress in Slovenia on next 16-17 October 2026. All of you are invited, and thank you so much for your attention.
Thank you, Professor, for your presentation. Surely, just to have a look what happens with leishmaniasis in Romania, we have data because we control a bit all the territories to catch sandflies, and using surely light traps or sticky traps like CDC recommendation was done, and till now we have not done more 0.1 sandflies in whole territory from 45 parallela through northern part and on south also. So in our faculty, in the last decade, we have only 3, 4, 5 cases of dogs who have leishmaniasis. 2 or 3 for them was imported, and the rest were autochthonous, we think. So a little question: if you tell us that in the next decade we will have spreading leishmaniasis in our territory, do you think just one health program could aid us just to not have more sandflies in our territories, because the climate changes, the highest temperature who could, ecologically speaking, could favorize the spreading of sandflies?
Yes, thank you for your question. Yes, I think that countries that are not endemic should be prepared with the epidemiological plan before that it will happen, because for sure it will happen. But, for example, the control for the importation of dogs, or to register the movement of animals when they go for vacation in endemic countries. When they come back, for example, they could be checked for serology if they acquired or not the infection. And yes, it is important to trap. But what is important in Italy, for example, that we have many data on sandflies because we can use the same traps for mosquito. So if you have the plan for West Nile virus, for example, or other mosquito transmitter diseases, in same trap you can also check for sandflies. Because this is another problem that sometimes there is a block of knowledge. Oh, I am studying mosquito, so in one trap I consider just mosquito and the other insect. So it's important to do a network all around the country to say, okay, also if your target is not sandflies, please think that this insect could appear.
Yes, thank you. We catched in our mosquito traps also sandflies, but for example, from thousands mosquitoes, we have 1 or 2 sandflies.
Yeah. But it's important to store also one sandfly to check for the species, because there are species that are able to transmit some species of Leishmania and others not. To check for PCR in this sandfly is positive or negative, could be could give good information.
It's not so easy to identify species by looking that on each one. It must be continuous by
But now with the new technology, you can take picture and send to people like in Italy or in other parts of the world.
Okay, thank you. You have other questions, please? No? If not, thank you very much.
Thank you, Ian.
We have a little bit late in this conference, so now we follow our program with an online presentation from Professor Nikolai Korcioni-Woski, Agri-Food and Biosciences Institute, Belfast, United Kingdom, from USV King Michael I Timișoara from Romania also. And this professor is the head of Food Microbiology and Agri-Food and Biosciences Institute in Belfast, Northern Ireland, UK. He is a fellow of the UK Higher Education Academy and elected member of the Royal Society of Biology. He is PhD supervisor biotechnology and teaching at several national and international universities. His research interests include bacterial virulence, isolation and testing of novel pre- and probiotics, development of novel technologies for a more efficient detection of foodborne pathogens. His research group is also performing research trying to understand the composition of gut microbiota and how this relates to the presence of bacteria posing a threat to human health, and especially in finding ways to manipulate the gut microbiota to reduce their presence. So if Professor, it appears, it is online. We ask Professor to enter for his presentation and to ask him to limit to 15 minutes if possible. Thank you.
Okay, can you hear me now?
Now it's okay. Thank you.
I'm not sure you can see my presentation. Second. So, thank you very much, and good morning, everybody. And first of all, just a big thank you to you all for this invitation. It's a shame I can't be there in your beautiful city, but actually, good afternoon to you all. I'm few hours behind you. But just to get through this presentation quickly, as I'm sure you're all thinking of the coffee break rather than just staying to listen to another talk, but hopefully I will try to excite you a little bit with my presentation. And basically, when I have this opportunity to be a keynote speaker in conferences, I take this opportunity to actually to more or less try to make a connections mainly with young scientists, and trying to ask them or to give them the courage to ask themselves questions. So my talk today on that line will be very multidisciplinary. So I'm going to be looking at how you can link after this umbrella of One Health that was mentioned before, how can you link climate change with, for example, microbial antibiotic resistance and animal health, and how can you mix those to actually uh, how can you mix those to improve the agri-food industry and to improve animal health and ultimately human health, because everything that we produce in the end ends up on the consumer's table. So all sorry my basically, like over the last few decades, my research was focused on the interaction between bacterial pathogens and the host, and trying to find interventions, for example, trying to find interventions to actually stop this interaction, finding, as I like to call them, smart interventions. Unfortunately, this movie doesn't work, but it basically just shows in few seconds you have bacterial pathogens that are actually attaching to epithelial cells. So it only takes few minutes between those two partners to actually interact, and that is again for the benefit of first of all. I think I'm on full screen. I'm not sure why. I think I'm on full screen. Am I? Can someone just let me know if I'm on full screen? Joys of presenting online. Sorry, can you see it now?
No, the share screen is off.
How about now? Okay.
We only see the presentation, not the share screen. If you can make share screen on all the screen.
Oh, okay, I know. Okay. Maybe now?
Perfect.
Yep. Okay. So just as I said, over the last few decades, my research was focused on this interaction with pathogens and the host itself. And you could see it now, luckily this movie works now, so you could see bacterial pathogens, in this case Campylobacter jejuni, for example, immediately attaching to epithelial cells. So basically trying to find and design interventions. How can you stop this interaction, and how can you avoid a pro-inflammatory effect? And this is all for the benefit and for the good of the agri-food industry, because you have healthier animals, you have less need to apply treatments like antibiotics, which we're trying to reduce massively their use currently. So this will be in the end for the benefit of all, and also for the benefit of the consumer itself. The reason we say that, because you see this, when these pathogens infect, and here you see the interaction between these Campylobacters, for example, and a human biopsy, and you see that the moment they actually infect, they actually interact with the host NADPH oxidases to produce this so-called oxidative stress. You could see them here in three-dimensional imaging. You could see them clearly interacting and triggering this NADPH oxidases to produce this hydrogen peroxide. And we have published many years ago this novel defense mechanisms in which clearly show that when Campylobacter infects these cells, they trigger this NADPH oxidases and they produce hydrogen peroxide, which is released in the luminal in the intestinal luminal space. This hydrogen peroxide is then able to actually dephosphorylate the tyrosine kinase in the outer membrane of these pathogens, leading in the end in dephosphorylation and post-translational modifications and some of the genes involved in bacterial capsular production. Because as we know, in many pathogens, these capsules are actually vital for virulence. So we published back then these defense mechanisms that actually suggested that things can be done to avoid colonization. So starting from that, and then introducing climate change, for example, why we do that? Because as we know now, climate change can affect many physiological mechanisms within bacterial pathogens, and one of them, for example, is their ability to resist to higher concentrations of antibiotics, or to completely resist. This is just a typical proof that indeed climate change has that effect. Also, we also know that climate change impact on virulence, regardless of the direction in which we move climate change. We move temperature, for example, we do have decreased variation if we go towards lower temperature, or if we go towards higher temperatures, we still have increased genetic variation, which leads to increased virulence in some of these pathogens. And I'm only giving Campylobacter here because it's one of the heavily microorganisms that we work in my research groups. And for that, just to stimulate the room and to stimulate the let's say the new people coming into this area of research, this is a typical example of a research project in which you actually take bacterial pathogens from farms, from hospitals, and from retail, because some pathogens, for example, do have they have this ability to infect humans, like Campylobacter. And the project, for example, could include isolation of these pathogens, and they can also then they can also go towards, for example, looking at infection models, and why not create a farm, for example, in which you can actually mimic climate change. So if we are getting into more and more trouble with these bacterial pathogens, for example, well, we should be able to ask, for example, answer these three simple questions in a way. For example, do we have alternatives? For example, can we use gut improvers? Can we use, I don't know, feed supplements or anything to improve gut health and to improve animal immunity? But most importantly, how do they work? The reason I'm asking this question might look simple, but it's actually not, because that's the problem that they actually faced back in the 40s and in the 50s, when antibiotics were used as growth promoters. Actually, Fleming himself, by giving an interview to the BBC in 1945, raises that question and tells us back in the 40s that be careful how much you use of this, because you might get in trouble later on. And we actually did. So are we actually using the necessary approaches, right? In regards to this antibiotic supplements, well, we just realized that we are actually in a way, now going back to the era of antibiotics and asking ourselves how that question, how they work. This is just a typical example how new alternatives work, for example, how essential oil, if you actually look at this slide and you keep in mind how an antibiotic works, you would actually be able to find out, that indeed these antibiotics, these alternatives, actually function in a very similar way. Most of these metabolic changes that you see here, they're actually able to be used in a similar fashion. And this is just an example. For example, this is just an example of one of my past PhD students. Actually, when she looked at Legionella, she tried to understand, for example, how organic acids as feed supplement work. So when she looked at drugs, she actually identified that at subinterior concentration, some of these feed additives, for example, or as we call them, organic acids or natural antimicrobials, you can call them that way. You can see they interfere with a lot of molecular mechanisms. First involved in iron sequestration, for example, which has a key role in biofilm formation in some of these pathogens, but mainly they affect bacterial motility and exopolysaccharide formation. These are all important virulence factors in bacterial pathogens and in biofilm formation. Why? Because we know that's also related to antimicrobial resistance. When she actually looked in vivo, she can actually see, she actually realized that it has an important effect in releasing oxidative stress and also in preventing the ability of these pathogens to actually infect these cells. The next approach and the next way to interact is, can we actually stimulate the gut microbiome to work with us to prevent infections, for example? And one of the interventions that people are now looking at, very popular in farming, for example, is the use of probiotics as alternatives. Well, this is easy probably in large animals. However, when we look at aquatic creatures, like for example, when we look in shrimp, applying probiotics in aquaculture is not easy. The reason for that the reason for that is because in this type of environment, giving probiotics is very, very difficult. Mainly because if you put this probiotic into feed, for example, some of them might get dispersed. So she actually showed really nicely that that actually you are able to use organic acids, for example, to stimulate a residing probiotic within the gut, within the shrimp gut, to prevent, for example, Vibrio parahemolyticus infections, which actually are devastating in farming in this situation. So clearly, they have this massive positive impact. And here you could see actually in these two graphs, you could clearly see that indeed they can prevent infection in these animals by only increasing the probiotic, this probiotic microorganism, Prausnitzia. From another PhD thesis that in the paper that we published, we also looked at other pathogens. For example, if we can mimic this approach to other pathogens, and we looked at Staphylococcus, we looked at the fecalis, for example, and in this case we looked at oral infections, and we looked at primary oral epithelial cells. And whenever you have an infection scenario, you immediately have a pro-inflammatory effect through this hydrogen peroxide, which is stimulating this external regulated kinase to cause an inflammatory effect and to cause this cytokine burst. However, when what we realized that when you apply interventions, like for example, when you add organic acids into the infection assay, you immediately mitigate most of this effect, and you have this significant anti-inflammatory effect. So clearly, this is just another typical example at molecular level how these alternatives can actually function. Similar impact we showed in parasites, for example. When parasites are infecting eukaryotic cells, they know to create this actin polymerization. Upon actin polymerization, they cause this increase in oxidative stress, antimicrobial peptides, and disruption of the epithelial barrier as its integrity. However, these interventions, these antimicrobials, this, for example, can be anything, can be even a plant extract, where we saw that this actin polymerization is prevented. We notice significantly less infection and a significant release in oxidative stress. So it has a really broad, but the molecular mechanisms in which they used to work are actually significantly different. And I would like to finish. By just showing you an example. One of my another PhD student of mine, who actually looked at Campylobacter within the UK poultry industry. What's the prevalence factors? Well, mainly looking at Campylobacter itself as a microbiome, and this PhD thesis was done in collaboration with Queen's University in Belfast, and Aaron here is the PhD student who produced this work. Again, I'm not going to go much to what Campylobacter is, but it's an important pathogen in humans. However, it resides in the gut of poultry, in chickens mainly, and it has no disease phenotype. However, what he noticed, and going back to this idea of climate change, and if climate change can affect these pathogens in any way they can. I mean, even if you only look at prevalence, and this is data that he analyzed for over 10 years actually, and he looked at over 350 million broilers. He works in a big company in the United Kingdom, and he was actually able to locate data from over 350 million broilers. And what he noticed was that indeed there's a variation in the levels of these pathogens that are actually present in this flocks. And moreover, he then published, for the first time, he actually was able to show. Here you see the microbiome in a chicken, in a broiler, and you could see that he was actually to describe for the first time the moment and the time when Campylobacter colonizes. You could see that approximately between day 14 and 16, you get colonization. By knowing this time of colonization, for us this was crucial, because it actually told us the time that when we can actually design efficient interventions, because we have also known by then the most important bacterial species that are present within the gut. So then he went even further, and then the story changed significantly, because he was actually able to design a study when we went really at high level, and we looked at 3 different production systems. We looked at microbiomes of these broilers in 3 different production systems: the regular productions, the so-called cheap chicken that you buy from the supermarket shelf, and then you have the higher welfare and the production system, higher welfare plus omega acids, so nearly in an organic system. However, what he designed, and I will finish my presentation with a video abstract of his thesis, but actually he was actually able to show really nicely and published in the journal Microbiome that you do have environmental factors that impact on the colonization and on the changes that you have in the microbiomes of these chickens. He was actually able to relate the stocking density to feeding, to types of feeding, and to the diet structures that are involved in changing the microbiome. in a different way. So basically, at the end of his PhD, we are actually able to say that if you have that ingredient in your diet, your microbiome goes that way, and you should be able to have that type of bacterial structures in your microbiome, which was quite significant at the time when he published this. So I like to hopefully this works and you can hear the sound, but I would like just to conclude by just asking you for 2 minutes just to have a look at his video abstract of his PhD thesis, and I can take any questions after that. So just to say thank you again, I'm not able to thank everybody in my group. That I'm not going to have the time to thank them all, but this is my research group here in a very descriptive way. But I'm sure they all know who they are. So thank you very much, and happy to take any questions if there are any.
Thank you, Professor Korczoniowski. If there are questions from audience, please.
Thank you, Professor, for being here with us, even via remote connection due to your health issues. Thank you for being part of our conference. I have 2 questions, very simple. Out of the production system you have presented in poultry, which is the most effective to prevent multiplication and contamination with Campylobacter? And also from the alternative methods you presented, what do you think it's also the most effective? Of course, apart from using the antibiotics as growth promoters because they are prohibited. Thank you.
I do apologize, but I'm just going to ask you to repeat your question. I couldn't hear a word you said, sorry.
Okay, I will resume. So what would be the most effective poultry husbandry system to reduce Campylobacter? And also, which is the most effective alternative method different from antibiotic to fight with Campylobacter?
So if I understood correctly, first you asked me second question was what is the most effective method to detect Campylobacter? Is it?
Yes.
Okay, so at the moment obviously at lab levels and institute level, we're using the ISO method for it, but we do have PCRs as well that we use for detection. In terms of interventions, if that is, and you can type that question on WhatsApp, and I can answer better because the sound is really poor. But basically, in terms of answering your question, in terms of interventions, I think at the moment we clearly know that Campylobacter is a commensal in the poultry gut. So at the moment, legislation in terms of Campylobacter is not asking you to get rid of Campylobacter, it's only asking you to reduce its levels to around 10 to the 3 in the chicken gut. And that meaning that most likely scientists now are debating this idea of actually Campylobacter being part of the regular digestive metabolism in these birds. We are asking ourselves, for example, a bigger question, and we don't know yet. How will a chicken without Campylobacter in its microbiome will look like? We don't have the answer to that question yet. I hope these were the 2 questions. Again, it's very good to hear the sound.
So thank you very much. There are more questions? If not, thank you again, Professor, for your presentation. And because it's the last speaker in this morning, I have an announcement for those persons who have vouchers paid. The lunch is on our cantina restaurant nearby the university, 5 minutes walking, till 2 p.m. Those who have not vouchers paid could pay on desk there. So after lunch, we will continue the program with presentation of scientific sections on each faculty. And don't forget to have on cantina your badge, and this on the back side of the badge you have the barcode where it's the inscription of your place and your work to be presented. So have a good lunch and have a good time. Good afternoon and see you late. Don't forget the last step to 7:30. The coaches are here inside our cart to catch you, to go to the restaurant for the dinner at Trajan Hotel. So there will be official dinner where you are all invited. Again, 7:30, the coaches are prepared. Thank you very much and good lunch.